So we have already looked into a scenario where certain regions in the PDB file, that is the extracrystallographic structure, was missing and were not resolvable. So there were specific residues that were missing in the N and the C terminal, and we had to reconstruct or build them by using the build tool in PyMo. So that's applicable for residues less than, let's say, 10 in number. Now, what happens for missing regions which are more than 10 residues, or let's say a large portion of the protein is missing, or for that matter, the whole protein is missing itself, which means the extracrystal structure was not resolvable at all for that protein, despite efforts made in that respect. So in this regard, we'll have to go for a different modeling approach. So it's a different scenario altogether where you do not have the structure of the protein available, but you have the sequence of the protein available. And you will find that the sequence is identical with an available structure of a different protein from a different organism, whatever that might be. So they might be identical, let's say 50% or 60% identity. But in order to perform this homology modeling, the percentage identity between the target sequence, which is the sequence for which the extracrystal structure is not deposited in the protein data bank, and the sequence for which a crystal structure is available in the protein data bank, that's called the template sequence or the template structure, has to be at least 30% identity in order for it to build a homology model of the sequence for which the structure is not available. So basically, these two sequences, the target sequence and the template sequence, are, if they match around 30%, or if they're identical, at least 30%, then they're known as a homologous sequence. Anything below 30% will not be amenable to homology modeling, and we have to resort to other methods, such as ab initio modeling, like prediction of the protein structure from the sequence itself alone by using threading or other forms of ab initio modeling methods, which we will see in the other tutorial as well. So the basic framework for homology modeling is first you obtain the sequence for which the structure is not available, like we'll do that via the Uniprot database, using the Uniprotkb database sequences here. And then we'll look for structures which are at least 30% identical, like in terms of the sequence identity, by doing a blast search against this target sequence. And we'll obtain those PDB structures, and based on those PDB structures as template, we'll model our sequence to its specific structure or fold that it might attain in terms of the secondary structures. And that will be our homology model. And so we'll get a solid prediction of the structure of the protein for which the sequence is available, but the structure is not available itself. Okay, so there are a number of softwares or packages, or let's say applications, that we can use to do the homology modeling. The most popular one being Modeler, which we have seen the web interface previously in the previous tutorial, where we reconstructed the loop regions and then we refined the loop regions by using the mod loop feature or the mod loop function or subroutine in Modeler by using the web interface. So it's available as a standalone package as well. But the problem with Modeler is that it uses the Python-based scripting language, for which some knowledge about the Python language is required. So we'll not go into that detail, and we'll do the whole homology modeling process by using a web server, a very popular one as well, and it uses some kind of a different algorithm than that of Modeler. Well, subtle difference between the two. And it's called the SwissModel, accessible via the XPaSI web server, and is provided and maintained by the Swiss Institute of Bioinformatics at University of Basel, Switzerland. But first, we need to obtain the sequence of the protein for which a structure is not available. And so the model system in this regard for this particular part of the tutorial would be the dopamine receptor D1. So as you have seen from the homework questions in the modeling tutorial, the homework assignment that I've given you, we went over a bit of the dopamine receptors. So it's a G protein coupled receptor, and it also is a transmembrane receptor. And we have specifically seen the domains in the dopamine D2 receptor. But surprisingly enough, the structure for the dopamine D2 receptor is available, whereas the structure for the dopamine D1 receptor is not available. The possible reasons being, since it's a transmembrane protein receptor, therefore it has a membrane component in it. And when you do the extracrystallography, which has to be done specifically in a lipid kind of environment, it might be sometimes difficult to resolve the structure of the protein in a lipid environment. So most of the structures in the crystallography method by crystallographers are obtained by uh, dissolving them in a water environment or using some crystal waters in order to obtain the structure. So, okay, let's go ahead and obtain the sequence, the primary sequence, the protein sequence for the dopamine D1 receptor first. So, as we type in here, the dopamine D1 receptor, and then press enter. So we are searching basically in the Uniprotkb database. And in the next results page, you'll see a number of options listed with the specifics of the protein that it got the hit for. So the first one here is the dopamine D5 receptor, and the source organism is human or Homo sapiens. And you can see the length of the protein displayed at the end column here as well. And the second option that you get is for the dopamine D1 receptor, the one which we are looking for, but the source organism is bovine here, Bos taurus, which is basically cow. As the third option is the one which we are exactly looking for, is the dopamine D1 receptor from the source organism Homo sapiens, and it has a sequence length of 446 residues. So we'll go ahead and click on this entry. Every time you see a Uniprot entry, it will be encoded with a specific accession code that belongs to the Uniprotkb database. So in this case, it's P21728. So you can commit to memory this code as well and have them as input for the blast search or even for the homology modeling by SwissModel. Okay, so you go and click on this sequence, and it leads you to a page with a number of information on the dopamine D1 receptor itself. So you have the function, and so the gene ontology annotations, like you have a molecular function, the biological process. It's a whole bunch of information that you can find out here. It's a very, very convenient database in order to look for specifics of a protein, be it whether the structure is available for it or not. It doesn't matter, but you will get a whole lot of information from here. So the location, especially where it is found in the body and everything, in the cell membrane and the endoplasmic reticulum and the membrane protein. So it's basically a membrane protein. So it will be found at the surface of the cell membranes. And you have the topological domain and the transmembrane domain. So there are 7 transmembrane domains here, and they're annotated by specific residue positions. It's extracellular and intracellular domain flanked by transmembrane domains, which are basically helices. If we go down further, scroll down, and we are basically looking for the primary sequence here. Now, as we stumble upon the structure feature, we find that there is a PDB entry, which is 1OZ5. But then again, the method of obtaining this PDB file, or the deposition, whoever the depositor was, has deposited it as a model, which is basically a homology model. Now, you have to keep one thing in mind, that previously, Protein Data Bank, like long back, let's say 5 years ago, it used to also accept models which were obtained by computational methods as opposed to the experimental methods. But recently enough, they have changed their course, and they're only accepting models which are obtained by experimental structure determination methods like X-ray or NMR or cryo-EM. And that makes sense as well, because, I mean, yeah, there will be a bunch of homology models that are generated through and through over the years, and all of them, if they get deposited in the Protein Data Bank repository, and it might be difficult to distinguish between the actual experimentally obtained structure and the computationally solved structure. So this is an obsolete record. As you can see from the resolution as well, it's not shown anything here because there's no structure available for this model. It clearly states here. Anyway, the PDB ID, the PDB code still exists, but we won't take this model into consideration, and we'll build our own model here. So we scroll down further, looking for the sequence, and then we come across the sequence. So you see the sequence, it starts from residue number 1 until 446. That's the total number of residues in the dopamine D1 receptor protein. We look for the FASTA sequence, so we click on FASTA. We obtain the FASTA sequence here, and so we make a copy of this. So we highlight whole lot of it. And then we copy this and we go and open the SwissModel page, which is swissmodel.xpasi.org. So you see the welcome screen here. It says welcome to SwissModel. It's a fully automated protein structure homology modeling server provided by the XPASI web server and the DeepView program as well. So we start modeling. So it asks you to paste the target sequence or the Uniprotkb accession code here, as I have shown you before, that you can also use the accession code to model the structure of this protein. So since we have copied the sequence already, so we are going to go ahead and paste it here. And it gives you a different representation, which means that it approves. So it considers it as a proper sequence. If it didn't approve, then it would give you an error. There's an option to add hetero target as well, which are basically maybe a metal ion or any other hetero atom or molecule that might come along with the protein structure. It might act as a ligand as well, like let's say a lipid molecule or something of that sort. But we don't have any sort of hetero molecule here. So the SwissModel also has the capability of modeling the hetero atoms or the molecules, but up to a certain limit. That is, it cannot recognize everything and anything. So it has a dedicated database from which it picks out information and can annotate specifically the heteroatoms or molecules as well. So, and on the right, you'll find supported inputs. So for us, the input is a sequence which is highlighted here. And then you have target template alignment. So if you know your template structure already, that is, you know what to model against, then you can have the alignment file of the target sequence with the template sequence as well. And then you can have an user template, that is the user submitted template structure, or if you have resolved the PDB structure and you want to test your unknown sequence with that of the PDB structure and see how they match up, and if you can build a 3D structure for your unknown sequence. And then there's this DeepView project option as well. So it asks you to give a project title, which is always handy in order for you to retrieve the finished job and access it properly. So let's say we give it a name of DRT1, which basically stands for dopamine receptor D1. And you can give your email ID as well. So once the job finishes, because this might take a while, depending on the number of protein sequences or the amino acid residues that we're going to model in the structure, it might take a while. So it's 446 residues. So it's better to have the email ID given. And when the job finishes, you'll get an email saying the job has finished. And in the meanwhile, you can do other things. So the first thing that will do is search for templates. That is, search for sequences which are at least 30% identical to that of this sequence and having a PDB structure, that is a properly resolved X-ray crystallographic structure against X-ray crystallographic structure for those template sequences. And the next thing that we can do is the build model. So once we obtain the template structure, we'll build the model of this sequence based on those template structures, which are homologous in nature to each other. So the first thing that we'll do is search for potential templates, that is the structures. So now the SwissModel template library is searched in parallel both with the BLAST method as well as another method called HHBits to identify templates and generate target template alignments. So the use of these two methods in consortium and generate excellent alignment of templates. So we go ahead and press on search for templates. We wait for the results to show in the next page. So as the program is searching for the templates, the respective templates that contains the structure with which it should model its sequence, it's basically showing you the information on whatever it's doing out here. So it's saying that it's running. So it might take a while depending upon the server speed and capability. And immediately it shows in what step of the process it is. As you can see, the lovely wavy, interpretation of the sequence as it is going through the sequence and trying to predict the template structures available against the sequences. So it says here running HHB lits against STML. So this is the library, and along with the BLAST alignment that it excuse me, along with the BLAST alignment that it incorporates in order to search for the template structures, and reusing cache query alignment and predicting residue burial status with ACC Pro. So it takes into consideration a lot of different features together, and then it tries to find the right kind of template or a number of templates that might be possible to model the sequence, the structure of the sequence that we have given as input. So it's running BLAST against SMTL now. And then it does a structural superimposition or superposition of templates as well. And then it also predicts the oligomeric state conservation. So it does a lot of things. And finally, it comes up with a list of 50 templates that it found hit against. Now, you have to take care of all these values out here. And what do they mean exactly? So these templates are listed according to their the coverage, the GMQE score, the QSQE score, which for most of the templates here is not shown, the identity percentage, the method that's used to resolve the structure of the template, and the oligomeric state of the template. That's basically monomer. In our case also, it's a monomer. And sometimes you can find oligomer as well, like in this case, it's a heteropentamer. And you have the PDB IDs and everything. So the scores, the GMQE score, it's basically the expected accuracy of the target template alignment. So the alignment of the target with the template is based on this score. So a score of 0.52 out of 1 is decent enough. It's pretty good, but it's the best you can get. And as you go down further, the score goes on decreasing. So these scores are calculated taking into consideration a number of factors. But out here, the information that you see that are taken into consideration are the coverage, first of all, that is how much of the sequence is covered by the template, like how much of a match does it create according to the residue spaces or the regions in the residues, and the identity percentage as well. Because for some, you will find that they have a high identity percentage, like 42.71, but the coverage is low, and therefore the the GMQE score is low as well. Okay. And so based on these two factors, the scores are calculated, and according to the hierarchy of the best template model that aligns with the sequence of the input protein that we have out here, which is dopamine D1 receptor, it ranks it accordingly. And so another thing that we got to look at is the QSQA. So you see here, it's not shown here actually, only for one of the structures, I think there's this QSQ score. So QSQ stands for quaternary structure quality estimate. So if you go down further, so for one of them, the score is given out here, 0.19. So it can't really estimate the quaternary structure quality for most of the template models yet. But finally, when we model it, it might give some information about that. So these are just predictions out here. The quaternary structure quality is just a prediction out of the innumerable considerations that we might have. Okay, so you see that the first selection is made out here, and we will only focus on the structures of the templates that are solved by X-ray crystallography. And also the resolution has to be taken into consideration to a certain extent, although a difference between 2.5 and 3.1 might not mean anything. But if we find a resolution somewhere around, let's say 1 point something angstrom, then definitely that's the first to go to for. But also we have to consider the ranking as well of the templates in that regard. Okay, so you can do a single template assessment to build the models, like align it with only one template to build the model. But since you want the modeling process to be very robust, you want to consider a number of templates into account in order to carry out the modeling process. So let's say we consider the first template, the very first template, which is solved by X-ray, the second template as well, which is also solved by X-ray, the third template as well taken into consideration. So as I'm going on adding these structures, it's going on updating it in the 3D representation out here and aligning each one of the template structures with each other. And you can see it's getting updated, the structural features. And the fourth one is electron microscopy, and the fifth one, so we are not considering the EM structure, but only focusing on the X-ray structures out here. So we can consider the fifth one. So you can see all of these are belonging to the same class of protein, the beta-2 adrenergic receptor, this the selection that we have made. So this as well, beta-2 adrenergic, this as well. So that's fine. That's all right. Like we are in the right direction because these gives the maximum hit with the same class of protein. And all of these have different IDs. That's because they might cater to different regions of the protein in the PDB structure. And therefore, if we model by using these four templates that we have selected, we might get a better match and build a better refined structure of the sequence that we have as input. So yes, you can see here that in the build models feature, you have four template structures selected, and against these four templates, we will build our structure for the sequence. So we click on build models. Again, depending on the capability of the server and how busy it is, it might take a while. So it says modeling job 3 is running, modeling job 1 is running, 4 is running, and 2 is running. So all of them are running together. Okay. So it brings you to the next page where they show you the model results and they rank it according to the GMQE score. So a GMQE score of 0.43 for the first model, and then a GMQE score of 0.44, 0.43, and then 0.43 further. But if you take into account only the GMQE score, then it won't be sufficient. So there's another global indicator of model accuracy, which is called the Q-mean. So a Q-mean score of 0 is actually an accurate prediction with that of the template structure, let's say. Whereas the lower you go, the predictions become worse. So a score below -4 would mean that that structure is not really well aligned and not really well predicted with respect to the template structure, let's say. So the Q-mean score for the first model we obtain is -4.32. So that shouldn't be taken into consideration. So we get a thumbs down for it, as you can see out here. Whereas the Q-mean score for all the other models, 2, 3, 4, against the four templates, are above -4. That is, they are somewhere around -3.68, but still not good enough. But I think that's the best we can get with these models because the sequence identity out here is barely over 30%. So the sequence identity above 50% would give rise to a better model as opposed to a sequence identity over 30%, but that's a prerequisite for homology modeling anyway. If it was below 30%, then we would have to consider other methods like threading or other ab initio prediction methods, let's say. Okay, so we'll not consider the first structure that we have here, which is basically model 3, modeled against the template 3 that we selected. And we'll go ahead and look at the scores of the second structure that we have here. That's a Q-mean score of -3.68. And it also gives you a local estimate based on the residue numbers and residue-wise propensities and how much they are fluctuating from the template structure. And they give you a comparative assessment as well. So since this is comparative modeling, so you'll get a comparative assessment. So your model lies somewhere around here with the star mark in red, whereas the z-scores, so the regions that are lighter have a z-score greater than 2, whereas the regions that are darker have a z-score less than 1. So the lesser the z-score, the better the model. So we can see that it barely fits into this darker region, somewhere around here. So we'll move on to the next structure and we'll expand it and find that the z-score again is not that great. So the model is lying at just an outlier outside the predicted region. If we go to the final structure, we'll find the same feature out here. So what we got to do is we got to focus on the Q-mean values now. So -4.32 we have already discounted, but within these Q-mean values, which range within -3, just above -3, we'll go ahead and do a selection amongst them. So the lower the Q-mean value, that is more closer it is to the 0 value, the more the value of the predicted quality of the structure. So 3.93, 3.82, so -3.68. is the way to go about it. So we select this model. So once we are happy with the model assessment, so we'll go ahead and do a kind of, so this is model 1 that we had selected, the very first model. We'll do a structure assessment. And in the structure assessment, you can see the Ramachandran plot, which you know already, you went through it, just to have a look at whether the secondary structure regions are lying in their proper places and there are no unnecessary outliers apart from glycine and proline. So you see they are fairly good enough and they're lying in their position perfectly fine. And finally, we'll go ahead and download the PDB. So we can download the coordinates here, and that'll be our model structure for the DRT1, the homology model structure, against the template of model 01 that we selected, which is a region of the beta-2 adrenergic receptor. So we download this PDB. It will ask you where to download it. So I'll go to my workspace directory, that's workspace 2, and give it a name, let's say model 01 drt1.pdb, and let's save the structure. Okay. So by default, the structure here, if I click here, it will open in Chimera. And you can see perfectly fine the 7 transmembrane helices, 1, 2, 3, 4, 5, 6, 7 transmembrane helices. And this is 19, which means this is the N-terminal region. So if we flip this on, and so this is the N-terminal region, and that's the C-terminal region. So the N-terminal region stays in the extracellular part, whereas the C-terminal region stays in the intracellular part. So yeah, that's about homology modeling with the SwissModel web server.